Again, it's a pleasure to be here, and I'm going to tell you a story of work that we started almost more than 10 years ago, but we have new results that have just recently appeared in the literature, and I hope I can tell you pretty much a summary of all that we have learned using methods of single molecule manipulation. I want to start by reminding you that in biology many processes actually are carried out by highly specialized machines that function, many of them, as molecular motors. Namely, they are able to convert the chemical energy, perhaps in the form of bond hydrolysis or concentration gradients, into mechanical work and force. We are interested in studying these machines, and in particular, we are interested in trying to understand how is that this energy conversion takes place, and what are the biological designs and the physical principles that underlie the process of energy conversion by these molecular machines. In particular, we are studying nucleic acid translocases. These are enzymes that catalyze a chemical reaction and move along a nucleic acid, and we use methods of single molecule manipulation to understand the operation of these machines. Today I'm going to tell you about one particular DNA translocase that belongs to the ASCII division of P-loop ATPases. And here is what biologists call a cladogram of the many different superfamilies that of these ATPases, NTPases, some of them are GTPases, that have the tendency to form oligomers, sometimes hexamers, sometimes pentamers, sometimes heptamers, and they carry a number of important functions in the cell. The one I'm going to tell you about is one that sits at the base of a virus, Bacteriophage Phi29, that forms a pentamer, and that whose function is to grab during assembly, the virus assembly, grab the DNA from the virus and pull the DNA and package it into the bacteriophage capsid. So here is a cryo-electron micrograph of one of these Bacteriophage Phi29 that we study. It's a small bacteriophage, icosahedral bacteriophage, only 40 nanometers by 55 nanometers. And at the base of the capsid, in fact, there is this molecular motor whose function is to grab the DNA of the virus after self-assembly of the capsid and put the DNA inside the capsid. It's an amazing task because in this very small volume you have to put about 20,000 base pairs of DNA. We know quite a bit about the motor nowadays. It's made up of 3 concentric rings, coaxial rings really. The first one, shown here in green, is the head-tail connector. It's a dodecamer. We know the crystal structure of this dodecamer. It was solved in the laboratory of Michael Rossmann in 2000. Then, surprisingly, there is a pentameric ring of RNA. So this is a piece of RNA that is associated, forms 5 molecules of RNA, form this pentameric ring in purple, and they surround the connector. And finally, there are the cylinders of the motor itself, which are essentially the ATPases. Again, there are 5 copies of this ATPase called GP16 that are the ones that are converting the chemical energy of ATP hydrolysis into movement. And so there is an opening through these coaxial rings that allow the DNA to actually get passed through into the capsid. As was mentioned this morning by Avinoad Ben Shaul, we have an experiment here that we essentially use to study this, how is that this packaging proceeds. We have a capsid that has started to package the DNA, but has been stopped from packaging all of the DNA, and then we put it on top of the surface of a sphere made out of plastic, a microscopic sphere, 1 micron diameter, 1.5 micron diameter, and we use antibodies to grab the capsid. And then the other end of the DNA, the distal end of the DNA, has a biotin, which is attached to a streptavidin, which is now on the surface of another bead that is in the optical trap. The optical trap is indicated here by the tick marks I show here. And now, in this situation, since this bead is atop a micropipette, as shown here, then we can essentially play tug of war with a single virus. When we add ATP into the solution again, the virus, the motor here at the base of the virus will start pulling the DNA, and we can move the pipette away or closer to the optical trap to make the packaging harder for the motor or easier for the motor. We can increase or decrease the tension against which the motor has to work. And then if you plot the distance between the bits, which is proportional to the length of DNA that has yet to be packaged, you see that the length of the DNA tether decreases in time as packaging proceeds. Here is shown for 4 different viruses, separated just laterally, so you can see, in fact, that the traces are quite robust, and it's possible to learn, for example, what is the rate of packaging from the slope of these curves, which are plotting length versus time. So the slope of the curves gives us the rate of packaging. Typically, we get packaging rates of the order of 100 base pairs per second, which is good because it is the rate that is believed to happen inside the host bacterium during infection. Okay, so let me just quickly summarize what we have learned through the years with this motor. First of all, it's a very strong motor, can generate forces as high as 60 piconewtons of force. This is very significant because myosin, which is the motor in our muscles, can, for example, generate only forces between 3 and 5 piconewtons. So this is 15 to 20 times stronger than myosin. The packaging actually leads to an internal pressure of DNA that was mentioned also this morning, and in fact we were able to estimate that internal pressure at the end of packaging, and you can see that experimentally very clearly, because if you plot the rate of packaging, shown here in the y-axis, in this plot, versus the percentage of the DNA that has been put inside the capsid by the motor, you see that after 50% of the DNA has entered the capsid, then the rate of packaging goes down, and eventually goes to 0 at about 105%. So you can put 5% more DNA in the capsid, but then the motor can no longer package anymore. Well, this corresponds to a slowing down of the motor because of the internal pressure, and that internal pressure arrives to a value of about 60 atmospheres. Okay? And again, this is a very interesting result because basically the problem the motor has to solve, the virus has to solve, is how to inject the DNA in the next cycle of infection into another cell. But now it's outside the cellular medium, there is no ATP. In fact, the virus doesn't even have the motor, because in order to put the tail, you have to take away the motor. So what the motor has done is convert the chemical energy of ATP hydrolysis into mechanical potential energy in the form of a loaded spring in the DNA at 60 atmospheres of pressure. Then when the virus sits on the surface of another bacterium, it uses these 60 atmospheres to propel the DNA inside the cell. So the solution that biology has found is a very mechanical solution to the problem of how to inject the DNA inside the bacteriophage, inside the bacterium during infection. We also showed that the power stroke, the moment in which the chemical energy is converted into mechanical movement, coincides with the release of phosphate. So once the phosphate has been hydrolyzed from the ATP, the moment the phosphate is released is where actually the movement of the DNA occurs. And we finally showed that the 5 ATPases that form the motor are not completely independent, but they are coordinated in some fashion. And we know that because we can do this packaging experiment and then add small amounts of non-hydrolyzable ATP. Non-hydrolyzable ATP will bind to the motor, and what we find is that the motor, in fact, stops completely as soon as one of the ATPases, one of the 5 ATPases, binds non-hydrolyzable ATP. If they were independent and one of them bound non-hydrolyzable ATP, you would expect that the other 4 could continue working, and the motor would not stop, it would just slow down. If you add a little bit more, maybe 2 of them will bind non-hydrolyzable ATP, and the other 3 will continue working. So the motor will slowly, will increasingly slow down. However, what you see here is that as you increase the concentration of non-hydrolyzable ATP, in fact, what you see is these very long pauses, and what you observe is that the pauses become more frequent but the motor always poses for a long time, for many seconds. In fact, we know that only a single ATP gamma S, in this case, which is the non-hydrolyzable ATP, is sufficient to stop the motor because the frequency of these pulses grow linearly with the concentration of ATP gamma S. So essentially, you only need one ATP gamma S to stop the motor. And that means that the 5 ATPases are coordinated in some fashion during packaging. They are not independent. So a model that we can imagine at this point, with what I just showed you of how the motor works, could actually be like this one, where you imagine there are 5 ATPases, and the ATPases work in some kind of ordinal fashion, 1, 2, 3, 4, 5. They bind ATP, hydrolyze the ATP, and when the phosphate gets released, which is the little star asterisk that you see here disappearing, you get the power stroke, right? Binding, hydrolysis, phosphate release, and power stroke. And we would expect that the DNA will be moved by about 2 base pairs for every ATP that is hydrolyzed. How do we know that? Well, people did actually bulk studies on this phage, and they found that on average 2 ATPs, 2 base pairs are packaged for every ATP consumed. Okay, so let me quickly show you some newer results that we have obtained recently, where we ask what is the nature of the interaction between the DNA and the motor that supports these 60 piconewtons of force, which is a lot of force, really. And so we imagined that maybe there was an electrostatic component, and in order to test that idea, we decided to modify the DNA that is being packaged by the motor here, and transform part of the DNA from phosphate to methylphosphonate. So we go from this form to methylphosphonate, which essentially preserves the structure of the DNA, but eliminates the charge in the DNA. And we did that, and in fact, what we found is that if you put 30 base pairs of DNA without charge as methylphosphonate, the motor, in fact, has a hard time going across the 30 base pairs. In fact, notice that if we apply 0.5 piconewtons of force of load externally, only about 10% of the time the motor can cross. If you apply 5 piconewtons, only 1% of the time the motor can go across the 30 base pairs. So this shows 2 things. One is that there is a very strong electrostatic component in the interaction between the DNA and the motor. But it also shows that it is not the only component, because if it would be the only component and you eliminate the charge, of course, we will get 0 here. But we don't get 0. We get a finite small amount of packaging, meaning that the motor must be making other contacts that are important during packaging, not just electrostatic. But then you may ask, does it matter which chain of the DNA has the charge, namely the Watson strand or the Crick strand? And you can actually test that idea by neutralizing the 3' to 5' strand in the direction of packaging, or by neutralizing the 5' to 3' in the direction of packaging. And when you do that experiment, you see that it makes a big difference. Whereas if you neutralize the 3' to 5' strand, then at 20 piconewtons of external force, you still are packaging more than 60% of the DNA. Here, if you neutralize the other strand, at 5 piconewtons of force, you are packaging less than 6%. So basically what this tells us is that the motor is making important electrostatic contacts, but with the 5' to 3' strand in the direction of packaging. Okay? Finally, we may ask, is there a critical length scale in which important phosphate contacts are made between the motor and the DNA? And we can do that simply by varying the length of the neutralized DNA from 1, 2, 3, all the way to 15. And you see that, in fact, there is not much significant change in the ability to go across the DNA when you neutralize in going from 5 all the way to 10. But when you go from 10 to 11, there is a big drop in the probability of crossing the DNA, of packaging the DNA, which we don't see again further on. So basically, what this suggests is that the motor is making important contacts every 10 base pairs with the phosphate in the 5' to 3' direction of packaging. It's very close to the pitch of the DNA. Actually, the pitch of the DNA is 10.4. But the 10 base pair, but it's important. It's a very important question, because at the end, I will show that there is, in fact, this discrepancy that needs to be resolved, and that leads to a new phenomenon that I will show you at the end. But so every 10 base pairs, there are these phosphate contacts that are very important between the motor and the DNA. I want to just say that one more thing, and is that we know now that this 10 base pair contact between the DNA and the phosphate not only has a mechanical loading function, but also a regulatory function in the motor. Because we can look at high resolution and see that when the motor is missing the 10 base pair contact, it doesn't even attempt to package the DNA. It's as if the motor doesn't know what to do. So this contact is important for the motor to package another segment of DNA. But this will become clear in a few minutes. Okay, so the next question we wanted to ask is, well, can we actually see, I showed you these traces where the length of the DNA gets shorter with time, but can we actually see the actual process of packaging, every step of packaging of the motor? In order to do that, we had to build an optical tweezer machine that has a resolution to 1 angstrom, basically at room temperature. And this is possible. It can be done these days. This machine probably has the record right now on spatial resolution. And in fact, you can put a bead. You use now 2 optical traps, 1 for each bead. We forget the pipette in this experiment to improve our signal to noise. And we put a piece of DNA, just for demonstration here, that is 3,400 base pairs between the 2 beads. We heard this morning that the DNA is a very elastic molecule, has a finite persistence length of 50 nanometers. And now you can ask, if I move this bead or this optical trap by 3.4 angstroms, which is the distance between one base pair and the next base pair in the DNA, can I record the movement of the other bead, even though part of that 3.4 angstrom is absorbed in the elasticity of the DNA? And the answer is, as you see here, yes, you can actually do that. And here are the curves that you obtained with this machine that allows you to measure 3.4 angstroms displacement of one bead relative to the other, which is one single base pair. You can do a pairwise distribution function of all the points in this curve, blue, or in this red curve, and you can see very clearly that we obtain enough signal to noise to recognize 3.4, 6.8, and higher multiples of a base pair. So with this instrument, we should be able to measure the step size of the motor as it packages the DNA in time. Okay, so let me now remind you what we expect to see. What we expect to see is that since we assume that there is 2 base pairs per ATP, what we expect is that the motor will wait for a while, bind an ATP, and then make a 2 base pair movement upon hydrolysis and phosphate release, and then again wait, 2 base pairs, wait, 2 base pairs, right? Okay, so this is what we were expecting, something like this. Binding, hydrolysis, and translocation. Binding, hydrolysis, and translocation. And that essentially we will see wait time or dwell time, translocation, dwell time, translocation, dwell time, and so on and so forth. This is what we expected to see. And we expected this translocation to be about 2 base pairs for every ATP. Well, we did the experiment, and as often happens, it's one experiment too many for the model. And so we, in fact, observed dwell times and burst and dwells and burst. But instead of seeing 2 base pairs, what we observe, regardless of whether we are below the Km of the motor or above the Km of the motor, all these curves here, all the way to 250 micromolar, the Km is about 40 micromolar, is that we have dwell times and then 10 base pairs, and dwell times and then 10 base pairs. So not 2 base pairs, but 10 base pairs. Okay? So this was quite surprising. We didn't expect that. We still observe the dwells and the bursts, but not 2 base pairs. In fact, you can do a pairwise distribution function for all those curves, for the different concentrations of ATP, and you see very clearly 10, 20, 30, 40, 50, etc. So the data is there. And now you may ask, well, how are these dwell times here distributed? You see that the dwell times sometimes are very short, sometimes are longer, sometimes long more, sometimes last very little. And so you may ask, how are these dwell times distributed what is the probability of observing a particular dwell time? Well, if there was a single ATP that was involved, that the motor is waiting here during a dwell time, just waiting for one ATP to bind, that would be one single stochastic process that would be controlling the length of the dwell time. And you would expect that these dwell times would be exponentially distributed, right? Well, what we found is that they are not exponentially distributed, which would be the black lines here, but whether you are below the Km or above the Km, you get these peak distributions. These peak distributions suggest that during a dwell time, the motor is waiting for more than one stochastic process, not just the binding of one ATP, but maybe the most simple idea is that it's waiting for binding of many ATPs, right? So one possibility is that perhaps the motor in Phi29 binds multiple subunits and then package in a burst. What I mean by this is the following. The motor has 5 ATPases, and maybe the motor is binding 1, 2, 3, 4, 5 ATPs, and then it goes 2, 4, 6, 8, 10, right? And these 2, 4, 6, 8, 10 are the 10 base pairs that we are observing. That was our hope as a way to actually solve save this model. Okay. It turns out that actually we decided we were going to test this idea. Notice one thing, by the way. I showed you that these dwell times are distributed as peak distributions. Here is the actual data for different concentrations, from 5 micromolar to 250 micromolar. If you increase the concentration of ATP, of course, the mean gets shorter, because it's faster to bind the ATP, but you always have this peak distribution. So we know that ATP is binding during these dwell times, and you may ask, well, what about binding of ATP during the burst? Well, you see that the burst is independent of the concentration of ATP over 3 orders of magnitude. So all the ATP binding is occurring during the dwell times and not during the burst. So that's important. So let me now tell you what is our packaging model at this point. We imagine that the packaging occurs in phases. The binding phase, which is during the dwell, right? And in this phase, the molecule binds N molecules of ATPs, right, as shown here. And then there is a burst phase where there is actual packaging. Here, we see here how the piece of the DNA gets shortened by this amount, 10 base pairs, during which, this is the burst phase, or packaging phase, during which there are N steps which correspond to N releases of phosphate. But of course the question is how many ATPs is N ATPs and how many steps are N steps. In order to answer this question, we can use our experiment if we were able to see the sub-steps that make the 10 base pair steps, right? So I told you that ATP doesn't bind during the burst, so I cannot slow down the burst by lowering the concentration of ATP. But what I can do is I can apply higher and higher load, force to the motor, and since this is the time where the motor is packaging the DNA, maybe I can slow down enough the motor in order to see the sub-steps, right? So the idea is to increase the force in the experiment so that we can see the individual sub-steps during the burst. So this motor is very strong. I told you that it can generate up to 60 piconewtons of force, so maybe we can slow down enough the motor to see the individual sub-steps. We did that, and in fact here is the data. We applied forces between 40 and 50 piconewtons, and now we are able to see these sub-steps. Very clear, sometimes you see one sub-step, but sometimes you see 3 of them together, sometimes you see 2 of them together. And now the dashed lines are not separated by 10 base pairs, but surprisingly by 2.5 base pairs. Okay, now that wasn't very welcome news for me, because as soon as my students told me, we know the answer now, in every step, in every burst, the motor makes 4 steps of 2.5, I said we will never be able to publish this because biologists don't like non-integer numbers. And so I sent them back to actually repeat the experiments. I wanted to make sure that the experiments were right, but they came back and showed me, in fact, that they could always see this 2.5 base pair. They did a pairwise distribution function. It shows 2.5, 5, 7.5, 10, etc. So it's very clear, the data is very clearly indicating that the steps of the individual steps of the motor are 2.5 base pairs. So, in fact, my students came back and told me, actually, we can tell you that it's 2.4 plus minus 0.1 base pair. So, this is the precision with which they can measure the step size of the motor at room temperature. 1 base pair is 3.4 Armstrong. 0.1 base pair is 0.3 Armstrong. So that's the precision with which they can actually measure this quantity. And incidentally, is what is possible these days with single molecule experiments. You can see changes that are very small. Since there are 10 base pairs in one full burst, it follows that there are 4 2.5 base pairs, right, that are involved in making 10 base pairs. And so you see that 10 base pairs are made out of 4 2.5 base pairs. So there are 4 steps and therefore 4 ATPs. So our model now is as follows. We assume that there are at least 4 ATPs that bind during the dwell time, and then you have 4 2.5 base pairs that constitute a burst. But that creates a new problem. There are 5 molecules of ATPase in the ring, and I'm telling you that there are only 4 ATPs that are being hydrolyzed into 4 2.5 base pair steps. So what happened to the 5th ATPase? So this answers the question of what is the model, but creates another problem, that there must be some kind of symmetry breaking mechanism in the ring, that otherwise, being a perfectly homopentamer, one of the 5 ATPases is not packaging the DNA by 2.5 base pairs. So this is a problem that remains, but I will answer that question at the end of my talk. Well, this is all data that is not published. Well, it's just been published, to be more precise, on the 22nd of November. This has appeared, and so I want to just show you that it's possible to learn more about this motor. For example, you may ask, where does the hydrolysis of ATP occur? I told you that during the dwell, the binding of the ATP occurs, right? So here you see, you can imagine that the hydrolysis can occur in 2 different scenarios. For example, the hydrolysis could occur during the dwell, immediately following the binding of ATP. So you get binding hydrolysis for the first ATPase, then binding hydrolysis for the second, for the third, for the fourth, and then you get phosphate release during the burst and packaging. Or you could imagine that the hydrolysis happens completely segregated from the binding, but also during the dwell. So you get binding, 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 binding during the dwell, and then hydrolysis, 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 and then the phosphate release during the burst. Or you could imagine that the hydrolysis actually occurs before the phosphate release during the burst. So can we distinguish between these 3 scenarios? And the answer is yes. You can actually use ATP gamma S again to stop the motor, and then you ask, where does the motor stop? During the dwell or during the burst? And in principle, you could answer the question, right? Okay, so we did this, but now we have high-resolution instrument. I told you that we built a very high-resolution optical Tweezers machine. So when we add non-hydrolyzable ATP, which is ATP gamma S, as before, you see these long pulses, and the frequency of this process grows linearly with the concentration of ATP gamma S. So we know that a single ATP gamma S is sufficient to stop the motor, all right? But now we see at high resolution what we mean by a pulse, by a single pulse. When you look at high resolution, you find that sometimes this pulse is a single pulse, as shown here, but many times the pulse is made up of, is really a cluster of pulses. You see here 1 pulse following by 10 base pairs, then another pulse. Or here 1 pulse, 10 base pairs, 1 pulse, 10 base pairs, 1 pulse, and so on and so forth. Now, the simplest way to interpret this data is you say, well, in some cases you have 1 ATP gamma S, in other cases you have 2 ATP gamma S. For example, 1 ATP gamma S binds, releases, and then the motor moves 10 base pairs, another binds, and then you have to wait until it releases, and so on and so forth. But that's not the case. In all these cases, these clusters of pulses grow linearly with the concentration, the frequency of this cluster grow linearly with the concentration of ATP gamma S, indicating that each cluster is produced by 1 ATP gamma S bound to the motor. Okay? Now, this is very important because we have another line of evidence. the length of a cluster, how long does the cluster last, which is indicated by this green light, actually decays exponentially in time. This length of a cluster is exponentially distributed, indicated that a single event, probably the replacement of a single ATP gamma S, ends a cluster. So we know that during this process, the motor has one ATP gamma S bound to it. And yet, the motor is not happy because it's not packaging fast. It's waiting seconds to actually package. And yet, every once in a while, it manages to make 10 base pairs. If it manages to make 10 base pairs, that means that actually the other 4 ATPases have ATP. 2.5, 2.5, 2.5, 2.5, right? And the fifth ATPase has ATP gamma S, right? So that means, immediately implies that each cluster is due to the binding of one ATP gamma S, and the motor can still function with one analog of ATP bound, and every once in a while the motor manages to make the 10 base pair packaging. So I'll come back to this because this is really important to understand how the motor works, but let me just answer the question about where ATP hydrolysis occurs. We are going to use ATP gamma S, and there are two scenarios. It can occur during the dwell, or it can occur during the burst. Let's assume that the hydrolysis occurs during the dwell, and that the first ATPase bound gamma S ATP. Of course, the motor cannot proceed in the dwell. It stops there. So that's the end of the packaging, and the motor has done this packaging, and then it stops there. If the first one, the first ATPase binds gamma S, and hydrolysis has to occur there. If it is the second, you also stop in the dwell, always in the dwell, in the third, in the dwell, in the fourth, in the dwell. So the important thing is that the last time you got a burst is always 10 base pairs in every case, regardless of whether it was the first, the second, the third, or the fourth that bound ATP gamma S, if hydrolysis occurs during the dwell. Instead, if hydrolysis occurs during the burst, you have the following situation. If the first ATP gamma S binds and is about to burst, the motor stops there and it cannot even do the first burst. So you get 10 base pairs. If it is in the second, the motor can make 2.5 because the first one can be hydrolyzed and release the phosphate and produce 2.5 base pairs. If it is in the third, you get 5 base pairs, and if it is in the fourth, you get 7.5 base pairs. So you can distinguish between dwell and burst for hydrolysis if you look at the size of the burst that precedes the beginning of a post cluster. So if you look at this burst before entering the big post with the 10 base pairs, you can tell whether it is in the burst or in the dwell. And we did that, and here is a demonstration of what we get. We get sometimes 10 base pairs, sometimes 7.5, sometimes 5, sometimes 2.5 base pairs. If you superimpose all of this data, using the previous bursts as anchors, the previous dwells as anchors, you see that what we get is a very broad peak that is not exactly a 10 base pair from the previous peak, because in fact you get 2.5, 7.5, 5, and 10 as an average. This is what you would predict theoretically if in fact the hydrolysis occurs during the burst, right? If the hydrolysis occurs during the dwell, you would expect very clearly 10, 10, 10, 10, 10 base pairs distance between these peaks. So in fact, we can conclude that hydrolysis of ATP occurs prior to the release of phosphate during the burst. I don't want to bother you with the details of this, but we can ask the question, where does ADP release occurs? ADP, once you hydrolyze the ATP, you release the phosphate, you are left with ADP, and you may ask the question, where does the ADP get released during the cycle of the motor cycle? And it turns out that crystallographers have a trick that is a very useful trick. You use a compound called orthovanadate, sodium orthovanadate, which looks like phosphate, binds to the ADP that is in the motor and doesn't allow the ADP to come out. So it actually essentially locks the ADP in the motor. We can use that technique, that same approach, and essentially ask, does the release R, here for release, of the ADP occur during the dwell, completely segregated from binding, or during the dwell, interlaced with binding, or during the burst following the phosphate release. These are the three possible scenarios. We use orthovanadate, sodium orthovanadate, to stop ADP release, and again, what happens is that the motor shows post clusters as before. So you see that the motor is forming these post clusters, and except that this time, instead of being a broad peak, you see that the peaks are separated exactly by 10 base pairs. So this means that the ADP release occurs during the dwell, the opposite of what we saw for ATP, occurs during the dwell. Of course, there are two possible scenarios for the dwell. The release may be completely segregated from binding, or it could be interlaced with binding, and we can distinguish between these two. Why? Because ADP is known to be a competitive inhibitor of the motor. In order for ADP to compete as a competitive inhibitor, it cannot be that all the release occurs completely segregated from binding. Why? Because in this case, it doesn't matter if you add more ATP, you could never outcompete the inhibitor because you have to wait until the last ADP has been released before you can bind the first ATP. So clearly, this model is inconsistent with the ADP being a competitive inhibitor, and the only possible mechanism is this. In fact, moreover, since you have to have all 4 ADP sites released before you can bind the first ATP, the ADP has many sites to bind to the motor, right? You have up to 4 sites to bind to the motor before the first ATP can bind. And you would expect, therefore, that the inhibition of the motor will be not linear with the concentration of ADP, but more than linear, second power, third power, fourth power in ADP. Well, you see that the inhibition is linear experimentally. So again, this is inconsistent with this model, and the only way you can reconcile the linear inhibition, competitive inhibition of ADP, is by assuming that there is interlacing between binding of ATP and release of ADP. Okay, so basically, we have this scenario for the release, and finally, I'm going to come back to the problem of the fifth subunit, because there are 5 ATPases in the motor, and there are only 4 steps of 2.5 base pairs. And now I'm telling you there must be some kind of symmetry breaking mechanism that is preventing one of the ATPases to function like the other ATPases. There is a special subunit, essentially, in the 5 ATPases. So let's go back to this observation of the post clusters. Remember, sometimes you see these clusters, and this cluster means that the motor has bound one ATP gamma S, and then the motor is not happy. In fact, the motor is stuttering because it's waiting for a long time, seconds really, 2, 4, 6 seconds, before it suddenly manages to make 10 base pairs, and then it stops. And then manages to make 10 base pairs, and then again it stops. So what is happening? We think that what is happening is that you have 5 ATPases, but one of the ATPases, its function is not to translocate the DNA, but to regulate the cycle. And normally, you have ATP binding to them, and all of them bind ATP. And probably all of them need to hydrolyze ATP, but one of them is telling the other subunit, start translocating. The other 4 subunits start translocating. What happens is when you add ATP gamma S, the motor is shocked because one of the ATP cannot be hydrolyzed, and so it ends up binding ATP to the other 4 subunits, and because it's waiting for this subunit to actually give them the go-ahead, the motor is waiting there. Eventually, spontaneously, the motor packages 10 base pairs, manages to hydrolyze the other 4 ATPs and get 10 base pair burst. And then the motor sits there again. The ATP is still, gamma S is still bound to this. It replaces it with ATP, and again the motor is waiting until to get the go-ahead for the other 4 subunits. But the motor cannot, because the ATP gamma S cannot be hydrolyzed, and it needs to hydrolyze in order to give the signal. So it waits there for a while until eventually manages to package again 10 base pairs spontaneously, and so on and so forth. Now, why is that we always bind ATP gamma S to the special subunit, you may ask. Of course, we cannot ensure that it. What we think it happens is that the subunit that binds the ATP gamma S ends up becoming the special subunit. It adopts the identity of the special subunit because it cannot hydrolyze the ATP and the other four can. And so the motor essentially is waiting for the signal doesn't come, eventually it does it spontaneously. Okay? But what that means is the following. This behavior of the motor is telling us that the motor has 5 ATPases, it needs to bind 5 ATPs, and it needs to hydrolyze the 5 ATPs, even though one of the hydrolysis is not being used for translocation of the DNA, but to regulate the cycle. Okay, so that's the model that we have. So what I'm saying is that the special subunit has probably a regulatory function, which is to trigger or resetting the mechanochemical cycle of the motor. And by the way, this model reconciles what had been observed in bulk, which shows that you have 2 base pairs translocated packaged for every ATP. Notice that if we assume that all 5 ATPases bind ATP and hydrolyze ATP, and you then end up packaging 4 2.5 base pairs, with namely 10 base pairs per burst, you have on average, in bulk experiment, you have 2 base pairs packaged per ATP that is being hydrolyzed, right? So this would explain that. Of course, this would not explain, you still may say, what breaks the symmetry of the motor? What makes the special subunit special, given that all 5 subunits are identical? There is a homopentamer, okay? Now, but let me just give you a rendition of what do we know now about the motor. The motor, the release of ADP during the dwell and the binding of ATP is interlaced, as I show here. Then the special subunit hydrolyzes and tells the next subunit start translocating, and then you get 1, 2, 3, and 4 steps of translocation of 2.5 base pairs. Now, here is a special subunit. I label it with S. The others are regular subunits. And now what happens is that if we said what happens during the dwell, the special subunit releases the ADP, and the binding of ATP produces the release of ADP in the next subunit in this fashion. And now the special subunit is the first one to hydrolyze and tell the others, without translocating, and tells the other subunit start hydrolyzing and translocating by 2.5 base pairs every time, as indicated here. Okay? So that model is consistent with the data we have about this motor. This indicates that the motor is very coordinated. The 5 subunits are talking to each other. There is an interlaced binding interlacing between binding and release of ADP, binding of ATP, release of ADP, and then there is one subunit that is special. But you may ask the question, okay, but what makes the special subunit special, right? Okay, so we found the answer to this question doing a completely different experiment that I'm going to relate very quickly here, and is where we ask the question, does the DNA rotate as it is being packaged into the capsid? Okay, so to answer that question, we have to do this experiment as before, where we grab the capsid of the bacteriophage. The bacteriophage is packaging, remember? But now we have 2 beads, one in the pipette, the other in the optical trap, and we put a tiny little bead on the side of the DNA, and we ask, does this DNA rotate, this bead rotate when the packaging proceeds? So does the DNA get rotated as packaging proceeds? Okay, so this is the experiment, and here is a movie that shows what we observe when we do this 3-bead experiment with the packaging of the phage. What you observe is this. As you can see, as packaging proceeds and the DNA is being internalized into the capsid of the virus, the DNA is being rotated. Okay? Okay, so the motor apparently can generate not only force, but also can generate torque on the DNA. Okay, so now instead of just plotting the length of the DNA, how the length of the DNA shortens as the packaging proceeds in time, I can show you now how the bead rotates, the angle of the bead rotates as the time proceeds. And in fact, we know now that the motor packages the DNA, it rotates the DNA in the direction of unwinding the DNA. So if you have the double helix, it tends to go in the direction of unwinding the DNA, and it can generate a torque of about 10 piconewtons nanometer, which is almost sufficient to denature DNA. So it's a strong torque generating machine. Except there is one problem. You can actually now do this as a function of the amount of DNA that has been packaged into the capsid, and you see that the amount of rotation per base pair of the motor, of the DNA by the motor, changes with the amount of DNA that has entered the capsid. And that's a problem. That's a problem because, let me show you here a small demonstration. Why is the problem? I told you, as we saw this morning also, that the DNA gets packaged in the capsid as a solenoid, probably, as concentric solenoid. So what happens if you have a concentric solenoid packaging inside the capsid? If the motor is doing this, and see what happens to the little rotator B in the middle of the DNA when you allow supercoiling to occur inside the capsid. See what happens to the bit? It's rotating. So I tell you the motor generates torque, and you tell me.
As long as you do it this way.
That's right. So
if you do it this way.
Well, if you do it this way.
I'm kidding.
Yeah. You'll always get supercoiling, so it will always actually do the rotation. So you may tell me, well, it's not that the motor is rotating the DNA. What is happening is that the DNA is getting organized as a supercoil inside the capsid, and in order not to plectonium outside, which is a higher energy form, the motor rotates through the single biotin-streptavidin bond in order to minimize the energy of the system, right? So, in order to convince you that the motor is in fact rotating the DNA, what I have to prevent is the DNA from organizing a solenoid inside the capsid. How do we prevent the virus to organize the solenoid inside the capsid? The answer comes from from something that I've seen since I was a child in Peru. The museums of Peru are full of these skulls where the Incas actually developed this technique in which the soldiers that had been actually damaged in their head during war had swollen brains, and that was solved by actually opening a hole in the capsid. And in fact, we know that many of these patients survived. In fact, here you see that this comes from this Journal of American Physical Anthropology article, because they put a piece of gold. They understood that the gold is the metal they should use in order to actually close the wound. And many of these patients have showed the bone growing over the gold, so indicating that the patient survived many years for the bone to grow back. So, in fact, it says that at some point it reached 90% of survival rate because they got really good at this. Of course, the gold is no longer here. The Spaniards took care of that a long time ago. In any case, the idea is, therefore, to try to generate trepanated heads. These are called trepanations, trepanated heads of bacteriophages. And we found that if we actually have the virus heads and we freeze them and thaw them and freeze them and thaw them many, many times, eventually the crystals of water generate holes in the head. And now we can actually do this experiment. And in fact, it works, because notice what happens when you have a regular bacteriophage head. You package length of DNA, plot length of DNA versus time. You see that the rate of packaging is slowing down, which is this slope, right? The slope gets slower and slower, the packaging rate, as the pressure of DNA increases inside the head of the bacteriophage. What happens if you use a trepanated head? You get a very straight line. There is no, obviously, because the DNA is going one way and coming out the other way. So we can then ask, under this condition, with these trepanated heads, what happens to the DNA? Does the DNA get rotated as it gets packaged? And the answer is yes. The DNA at zero filling gets rotated by minus 1.5 degrees per base pair. Okay, minus 1.5 degrees per base pair, which means that some of this higher rotation is due to the organization of the solenoid. But at zero packaging, the motor still rotates the DNA. And what is minus 1.5 degrees per base pair? And here comes the question that you asked just a few minutes ago. It turns out that I'm telling you that there is one subunit that is a special subunit. I'm going to tell you now and propose that that subunit is a subunit that contacts the phosphate every 10 base pairs. Remember, there is a phosphate contact that is made every 10 base pairs. Let's assume that that contact is made by the same subunit, and that subunit, as soon as it makes the phosphate contact, becomes the special subunit. It knows that it should not translocate, but it should regulate the other four subunits. So that subunit made the contact now, then 2.5, 2.5, 2.5, 4 times 2.5 base pairs are packaged, and that same subunit wants to contact the next 10 base pairs, the next phosphate, 10 base pairs afterward. However, the pitch of the DNA is not 10 base pairs, but 10.4 base pairs, which means that while the motor has advanced 10 base pairs, the DNA phosphate has not rotated 360 degrees, but has rotated only 346 degrees, because it takes 10.4 base pairs in order to rotate 360 degrees, which means that that phosphate is 14 degrees away from where it should be. So the motor, every 10 base pairs, rotates the DNA by 14 degrees in order to make that special contact. And so 14 degrees in 10 base pairs is 1.4 degrees per base pair, or 1.5 degrees per base pair, which is what we measure. So the model that I'm proposing is that the motor makes a contact every 10 base pair with the phosphate through the same subunit, which is the special subunit, which doesn't package, just regulates. And then after 10 base pairs, it needs to correct the difference between the 10 base pairs and the actual periodicity of the DNA, which is 10.4 base pairs, and that gives rise to the rotation that we observe at zero filling. Okay, so that's the idea. Essentially, there is this 14 degrees needs to be corrected after 4 10 base pairs, 2.5 base pairs packaging, and that means that you have to go from 346 degrees to 360, and these are the 14 degrees per burst, or minus 1.4 degrees per base pair, which is pretty much what we measure. We measure 1.5 degrees. Okay, so let me now, this is the last slide, let me now give you a movie that actually puts together all of this model, what we know about this model. What you are going to see is the 5 ATPases, and one of them has a different color because it's the special subunit, and is the one that makes the contact with the phosphate every 10 base pairs. Okay, so here it is. The motor is ejecting the phosphate and translocating the DNA. See, here it starts. This is the special subunit. Gets rid of this ADP, binds ATP, that releases the ADP from the next one. Binding of ATP releases the ADP from the next one. Binding the ATP releases the ADP from the next one. And this is the special subunit. It hydrolyzes first the ATP, but doesn't translocate. It tells the other subunits, start translocating and hydrolyzing your ATP in this fashion. And then at the end, the special subunit needs to make the contact, so it rotates the DNA by 14 degrees. And so again, a new cycle starts. Binding of ATP releases the next ADP in an interlaced fashion. Binding releases ADP, and then the special subunit hydrolyzes again first, but doesn't translocate, and tells the other ones hydrolyze and translocate, hydrolyze, translocate, 2.5 base pairs every time. And at the end, the special subunit needs to rotate 14 degrees in order to actually make again that crucial regulatory 10 base pair phosphate contact. So this model is consistent with everything that we know about this motor at this point. It may not be right, it's probably not right, but actually it's as right as it can be at this point.
If there is a slip, then you move to another one.
Yes, exactly. In fact, there is nothing special about the special subunit other than the fact that it's the subunit that makes the contact and that breaks the symmetry of the ring. So many times while we are packaging, and particularly when we apply a lot of tension, in fact the DNA actually slips a little bit. And in that case, the motor stops for a while, and pretty soon it starts regulating itself again, because one of the subunits makes the crucial contact with the phosphate, and it becomes the special subunit every time.
But that means that you can actually make some predictions about the rate that when it decreases at that high pressure, when you try to compress more and more the DNA inside because it doesn't go to 2.5 to 4.5, so this is linear, right? That should be going on the same rate for no matter what force it is. I mean, the other force from the attacking of the DNA actually slips it back, right?
No, so what happens when you have internal pressure, so that's a good question. In other words, so what happens when the motor slows down? So does it mean that the size of the burst gets smaller, or it means that something takes longer? And what we observe is that what it means is that actually the dwell time gets longer but the motor still base 2.5, 2.5, 2.5, 2.5. So the 10 base pair structure is maintained, but what happens is that it takes longer time between the individual sub-steps or during the dwell times. So that, on average, that means that the slope of packaging gets smaller and smaller. That's a very good question, because in fact, I show you that when we apply external force, what we do is we get the sub-steps longer so that we could see the measure of the 2.5. When we package 90% the genome of the phage, this also happens. But in addition to that, the dwells get longer. And we think that actually that's a mechanism of regulation also for the DNA to organize itself, that it slows down, that the pressure slows down the motor at the dwell, not only at the birth, but at the dwell, in order to perhaps allow the DNA to find itself inside the capsule of the virus, to arrange itself in the capsule of the virus.
But the dwell time is all dependent about the chemistry of hydrolyzing the
binding of ATP, release of ATP.
Yeah. So all of that is outside. How do they know what is needed from the DNA from the inside?
Well, I think that actually the signal is the pressure, the hydrostatic pressure that is reaching almost 50 atmospheres, 60 atmospheres at that point. And that is really affecting directly the operation of the motor. So this is not published, and this is data that we are really gathering right now, but I can tell you that the internal pressure slows down the burst, but also lengthens the dwell time. Whereas the external pressure only slows down the burst, but doesn't affect the dwell time. So the internal pressure must have a special kind of effect for the motor, which we think is perhaps regulatory at some point. Yes.
So in this model that you presented, rotation is uncoupled from translocation.
No. In fact, that's a very good question. Namely, you could imagine that every time the DNA gets translocated by 2.5 base pairs, it gets rotated a little bit, which is perfectly possible. We don't have the time resolution in our angular experiment to decide where is that the actual rotation occurs, whether it's during the burst or at the end of the burst. So your point is well taken. We assume that it's at the end because it's easier to demonstrate in the model 14 degrees than 14 degrees divided by 4, which is 2.5 or 3.5.
I suggest that we just let Carlos finish the lecture and then there will be time for questions. Pretty much to the end.
Yeah, no, I have to say the people that have done the work, 4 great postdocs under task with Sackler, Netherlands now, Dan Smith, a professor at UCSD. Jan Chandra is a professor of physics, also at the University of Illinois, Champaign-Urbana, and Ariel Kaplan, who is here, is a professor at the Technion. 4 graduate students that have done really great work. Attica Lucaran is now doing a postdoc, did the work with the phosphate neutralization. Jeff Moffitt determined the 10 base pairs and the 2.5 base pairs in between. He's at Harvard with Xiaobo Zhang. Ati is with Ron Bay at UCSF. And Craig Hetherington and Georgi Keystone are still in the laboratory. Craig did the work of the rotation of the DNA with the Trepanated Heads, and Georgi did the work with the ATP hydrolysis and ADP release that I showed you. And Xinxin Liu is the postdoc from Xiaobo Zhang's laboratory again, that is really working with Georgi in this experiment. The work was done in collaboration with the group at the University of Minnesota, Roy Anderson, Shane Grimes, and Paul Berkeley. So that's
there is now enough time for all the questions. Yes, please. Yes.
That is correct. All subunits have abilities are identical. So the selection of the so-called special one is random. And in the case of the ATP gamma X, if it binds and then it becomes, eventually you get four steps. So suggesting that the so-called special one is the one with ATP gamma X.
Yeah, that
how does it manage to get into the next step without being hybridizing the ATP? It can jump the 10
Yes, exactly. So the model is extremely coordinated, 5 subunits. And normally you require the hydrolysis of the special subunit in order to start hydrolyzing and translocating. When that doesn't happen, the motor is waiting there for eons in molecular terms of time, because it's there 2 seconds, 3 seconds, 4 seconds, and then eventually, spontaneously, it manages to fire, and all the 4 subunits fire spontaneously.
Yeah, but what about the fifth now?
The fifth subunit is still bound to ATP gamma X. The ATPs apparently can be exchanged, and as long as the ATP gamma X is still bound to that special subunit, still will happen the same thing. The others will replace the ATP.
Jump the 10 bases each time.
Yeah, because you need 4 ATPs, and there is plenty, there is excess of ATP in the middle. So
will it be possible to make it for covalent unit and compare with one subunit effective in the water A, for instance? It was done by Sackler.
we have mutants now. The important thing now is to try to generate a single polypeptide chain for all 5 subunits, like Bob Sauer. That's right. And in fact, we are working right now with XT, which is because Andy Martin, who worked with Bob Sauer, is down at Berkeley. So we are collaborating with XT. We are using the pentameric, the hexameric ring in the single polypeptide. Whether it's possible to translate that from QTXT to the Q39, we don't know. We are going to try. In order to make a mutant in a particular one of them. Or 2 of them. That's right.
Other question?
You measure the force, and you know the displacement. And I guess we know the energy each per ATP.
Yes.
So what related to the 3 of them. So
What is the efficiency?
well, that's one question. That's a continuation. I mean, is it like a reversible process? Or what is the efficiency?
Yeah, so let me start with efficiency. We know how much energy is in an ATP, and the conditions in which we put is about 12 kilocalories per mole, but we translated into piconewtons per nanometer is about 100 piconewtons per nanometer. And we know the maximum force that the motor can generate is 60 piconewtons, and we know it's 2.5 base pairs per ATP. That's almost 8 angstroms. So that corresponds to an efficiency of about 62 to 65%. So it's a very efficient motor. These motors are really, really efficient compared to human-made machines.
But this takes into account the fact that which one doesn't make the transfer.
Yeah. Because you say,
close to waste.
yeah,
Or
10 base
maybe
pairs.
just for the twist.
Yeah, because you do it per ATP. So for 4 ATPs and 10 base pairs and 60 piconewtons, you can calculate per ATP. So let's assume 1 ATP. The allocation is 2.5 base pairs per ATP against 60 piconewtons. So that's the work done by the motor. And we divide that by the energy in an ATP, in the concentration of ATP and phosphate and ATP in the medium, and that gives us 62%. That's when we lose 60%. So it's very efficient. Now, is the motor reversible? That's a very good question. In other words, can we actually pull the DNA through the motor and make the motor synthesize ATP instead of consume ATP? Like reverse it.
This was your idea, not my question.
This is your question because if it is reversible, it should be possible to do that. We are trying to do that experiment. But the idea is to pull the DNA, to put excess phosphate and excess ADP, and have the motor carry out a reverse reaction and pull it mechanically to help it. And every time you synthesize an ATP, you put some luciferase in the medium, and it produces photons. And we will see the photons coming to us.
Thermodynamics.
But it hasn't worked yet. It's right now. I cannot tell you that it's reversible.
And what we need to try to do is experiment, for example, in the presence of an interconnector, something that will change the piece of the DNA so that you can figure out which is the motor property of the species or rather related to the DNA.
Very good question. Excellent. In fact, there is nothing in the DNA that has a releases of 2.5 electrons or 2.4 electrons. Nothing in the DNA has that. So what we think is happening is that the 2.5 electrons that we observe is the size of the conformational change that the ATPase undergoes and the release of photons. Namely, when the ATP binds, the ATPase cuts itself and leaks the level by 2.5 electrons. When it hydrolyzes the ATP and releases the phosphate, it goes back to its initial position, and that's 2.5 electrons. Now, I told you that every 10 base pair there is a phosphate contact. It's clear from our experiment. What happens in between? Because in between is that the actual padding is happening on the DNA. We think that those contacts are not electrostatic but rather state relation. Why do we know that? Because we can, in fact this is published, we can replace by an aircraft. We can actually take the bases out of the DNA and just use the phosphodiester backbone and the motor chip packaging. You can actually replace completely the DNA by a polymer that looks like polyethylene glycol and the motor chip packaging. So the motor is very promiscuous between the 10 base pair, the phosphate contact, that requires the phosphate contacts in order to be regulated, as I indicated here. So in fact, it's a very promiscuous motor, and if we put a bunch of DNA, one base extra in one of the strands, still packages. So it's very, very promiscuous in between the phosphate. The only thing that it really requires is a 10 base pair phosphate contact in one of the strands, the 5' strand.
What happens in the rotation in the presence of the ATP-vanadate? So particular ATP-vanadate, does the motor still rotate?
We haven't done this experiment, but the rotation experiments haven't been published yet. We are still writing the paper, and we haven't done it in the presence of ATP-vanadate. But I suspect that what happens there is that the motor stops, and the rotation has to stop, because it's clear that the two are coupled. Whether they happen at every sub-step, or whether they happen at the end of the 10 base pair burst, there is a coupling between rotation and translocation. So if you stop the motor, I imagine that you will see the bead stop rotation. And we don't have the resolution yet at this point to tell whether it happens.
You still have the spontaneous translocation.
During the cluster, you said. During the cluster, yeah. Yes, but we haven't done this version yet. So it's very slow, remember though, right? You get 4 seconds, 10 base pair, 6 seconds, 10 base pair. So you will see very, very little. Probably the noise in the bead, we need to make the bead much smaller. So Seth Bryant, who is at Stanford, where I sit in my lab, now has developed a much higher resolution rotation for different systems. The rotational assay, and I think with that, which is almost 50 nanometers beam, it's possible to get a much better time resolution for rotation. I think that's what we need to do in order to do that experiment. The good idea. These are some of these experiments are fun.
Another question. Yes. So maybe just as a way to kind of resolve this, I don't know if it's possible technically, but just to immobilize the actual DNA strand and then see how the motor itself will twist on and immobilize the DNA strand.
Yes. In fact, with the trepanated head, you could imagine the whole head to rotate around that's right. And maybe we could actually, for example, put a fluorescent label and then look at the polarization, fluorescent polarization, and follow the rotation by the fluorescent polarization. So that would be another way to do it, yes. Yeah, it's possible. Again, these experiments are tough, but they are not so simple. In principle, it's possible, I think.
Other question? If not, let's thank Carlos again. I would like to close this today by, first of all, congratulating again Carlos and Wolfgang for the prize, and saying that we at Tel Aviv University are very honored to host such an event. I think, and I'm speaking especially for the students, I think that one of the take-home messages is that great science come in all sorts of shapes and colors, and there is no unique way, no recipe how to do it. And I think that the main thing is the inspiration that we could get from the two speakers and the two laureates. And I also equally would like to thank very much first Sackler for the interesting introductory remark at the beginning of the morning session, and then the 6 speakers, some are own from Tel Aviv University, others from Jerusalem, and 3 from China and United States and Germany. I think it was a very nice session in the morning, and we heard very interesting science in biophysics, and I would like to conclude here, and again, thank you for participating. Thank you.